Welcome back to the Answer Series Diagnostic Report Teaching and Learning Tool for the NSC November examination. This is an all-in-one tool that combines the November NSC paper, Memoranda, and annual Diagnostic Report commentary to create a comprehensive learning experience. TAS provides this document for free, and we encourage teachers, learners, tutors, and subject advisors to use it. All we ask is that you please give credit where credit is due and not change or alter any content in this document that removes our name from it. Question 2.1 has to do with protein synthesis. The diagram represents a certain stage of protein synthesis, and the sequence of the bases is read from left to right. So they ask us to identify molecule Y. If we unpack the diagram, we see molecule X is a helix structure, and there's a section of it that has unwound to form a strand 1, which is the template, and a strand 2, and then molecule Y is a single-stranded structure that's being constructed on top of strand 1, the template. Molecule Y also has the nitrogenous bases A, G, U, and C, and uracil is present in RNA. So this is mRNA because it's the only RNA that associates with the DNA directly during transcription. Then question 2.1.2 asks to describe the process that resulted in the formation of molecule Y. So this step in protein synthesis is the transcription stage. They're basically asking learners to rewrite what is written on the exam guideline that provides a very clear description of transcription. So the first mark is awarded for the double helix of the DNA unwinds, then the concept of unzipping or the hydrogen bonds breaking. 2 separate strands formed. One of these is used as a template to form an mRNA strand using free RNA nucleotides from the nucleoplasm, and the mRNA will be constructed complementary to the DNA, or a learner could have said that for every A on the DNA, a U is added to the mRNA strand, and for every G on the DNA, a C is added. So candidates could not identify the process represented, so they struggled with understanding the picture, and therefore they gave a whole description of DNA replication instead of transcription. Candidates lost marks for omitting important information like a DNA double helix, talking about RNA nucleotides, and that only one strand of the DNA is used as a template. So it's really important to help learners focus on a diagram like this and unpack it before approaching the question. This picture shows transcription because we can see 2 different molecules. So we can see molecule X and a molecule Y, which tells you that it's not only DNA in this picture. We can see a transcription bubble, meaning that the DNA has unwound in a specific section, creating a little bubble in which the transcription occurs. So it's important to look at the pictures of DNA replication and transcription next to one another so that we can identify the differences between them and help learners identify them in exams. Then question 213 asks for one structural difference between molecules X and Y. So having identified molecule X as a DNA molecule and molecule Y as an mRNA molecule, we can now compare them. We revised this in the first video for paper 2's diagnostic report with a single-page summary of the similarities and differences between DNA and RNA, and you can go to that video for that discussion. So the list here includes that molecule X, which is the DNA strand, is double-stranded. It's a double helix, whereas molecule Y is single-stranded. In other words, it's not a double helix, or it doesn't have a double helix. Molecule X, the DNA has hydrogen bonds. Molecule Y does not have any hydrogen bonds. Molecule X contains deoxyribose as its sugar. Molecule Y, the RNA, contains ribose. DNA has thymine as a nitrogenous base, whereas the molecule Y has uracil. And then molecule X is longer, molecule Y is shorter. So there are many structural differences that they could have highlighted. Any one of these structural differences times 2 marks. And importantly, because the instruction says to give one structural difference, a marker is instructed to mark only the first difference that has been given on the script. Question 4 asks us to give the sequence of bases at triplet Z on strand 2. So there are 2 ways to do this. The one way is to take the long route. So first, you have to look at the mRNA molecule. Then UAC needs to be transcribed back to what was on strand 1. So UAC on strand 1 would have been ATG. and then strand 1 and strand 2 are complementary. So ATG on strand 2 of the DNA molecule would have been TAC. The other option is much shorter, which is to notice that if molecule Y is being transcribed from strand 1, and strand 1 and strand 2 are complementary, then strand 2 will follow exactly the same sequence as molecule Y. It's also the complementary copy of strand 1. And the only difference is that because it's a DNA strand, any U's will be T's. So the sequence is TAC. Candidates lost marks because they gave the complementary base sequence for strand 1 instead of that for strand 2, where triplet Z occurs. So they either misread the question or didn't understand how to work between the two DNA strands when there's one mRNA strand. Question 5, the table shows some amino acids coded for by codons on molecule Y. So it's very important to note that what's given in the table are mRNA codons. Identify the first and the last amino acid coded for by this section of the molecule Y. So it basically means that we have to read from left to right as the instruction in the question initially stated, and we have to take every 3 base pairs to be a codon and then translate that into amino acids. So AGA is the first codon sequence. In terms of the table, that translates to an arginine amino acid. Then they don't ask for UAC, the middle codon. They're only asking for the first and the last amino acid. So the last codon is CCA, and CCA, according to the table, codes for proline. So very important to read from left to right to get the first and the last amino acid. Candidates were unable to interpret the codon table and its relation to the diagram. They missed that the stem of the question indicated that the sequence of the bases should be read from left to right. So that implies that if candidates first wrote proline and then arginine, they would have lost the 2 marks because a marker would have interpreted this as proline being seen as the first amino acid and arginine as the last. So learners really need to pay close attention to the stem of a question that often provides information about how to work with the question. Then question 2.2 is about meiosis. The diagram represents a phase of meiosis 2. So very important, we've already halved the number of chromosomes because meiosis 1 is the reduction division and genetic variation has already been introduced. Question 221 asks us to identify the phase of meiosis shown. So half the thinking has been done for the candidate in that they know that there should be some phase within the second part of meiosis. Now, if we look at the diagram, we see that there is a separation of chromatids or unreplicated chromosomes moving to the poles. The spindle fibers are shortening. The chromatids are making these little boomerang shapes, which means they're being pulled towards the sides, and that is typical of anaphase. So anaphase 2. The diagnostic report says that correct application of biological terminology was vital in this question, and candidates in question 221 lost marks for providing only anaphase instead of adding the 2 with it, which is really sad because it's actually stated within the stem of the question, so half the answer was there. Then question 222 says to state one difference between the phase that is shown in the diagram and the same phase in meiosis 1. So how does anaphase 2 differ from anaphase 1? Now we revised this again within the first video of paper 2. We had a nice single-page summary of the difference between meiosis and mitosis and the different phases of meiosis. So in terms of anaphase 1 and anaphase 2, the difference will be for anaphase 2 in this regard that chromatids are moving to the poles, whereas in anaphase 1, we would have whole chromosomes moving to the poles because the pairs of chromosomes are being separated in anaphase 1. Another difference is that in anaphase 2, as we can see here, the centromeres are being split in order to pull the chromatids apart, but in anaphase 1, because the whole chromosome moves to the pole, no centromeres are being split. And again, the marker instruction is to mark only the first difference that is provided as the question asked to state one difference. So again, the diagnostic report talks about the biological terminology that was a problem in question 222. Candidates used chromosomes, chromatids, centromeres, and centrioles interchangeably. Very incorrectly confused them all. And they did not distinguish between homologous chromosome pairs that separate in anaphase 1 and single chromosomes that separate during anaphase 2. There are, however, two ways to have stated this, as you can see by the dash. So learners could have said that chromosomes, whole chromosomes, move to the poles compared to chromatids that move to the poles. Otherwise, they had to compare pairs that are separating during anaphase 1 compared to no chromosome pairs. Just a single chromosome separates during anaphase 2. Question 2.2.3 asks us to identify structures A and B. So structure A shows us a centriole at the pole. Learners would also have gotten the mark for saying centrosome, which is the organelle in which the centrioles are located. And then B is the structure produced from that centriole, which is a spindle fiber. So candidates wrote centromere and confused that with centriole or centrosome, so we know that those words are confusing. In question B, candidates wrote springle or spingle or sprindle fibers instead of the correct spelling of spindle fiber, as we noted in the first video of this paper's Diagnostic Report teaching tool. Question 224 says to describe the role of structure B during cell division. So what is the spindle fiber's function? So its function is, if you just look at the picture, you could have actually described this. It attaches to the centromere of a chromosome so that it can pull the chromatids apart and towards the opposite poles. Learners could also have said that it contracts or it shortens so that it can pull chromatids or unreplicated chromosomes towards the poles. So 2 marks for that. Then the diagnostic report mentions that candidates used colloquial terms like sides or ends instead of referring to the actual structure, which is the pole. So they said that it contracts and pulls chromatids to the sides. Again, life sciences is a language. Biological terminology needs to be taught. Learners need to speak the language, just like any other language, German or French, that you want to get marks for in an exam. Question 2.2.5 asked to draw a labeled diagram representing this cell as it would have appeared in metaphase 2. In other words, just one phase before this one. And then they tell learners that we're not going to penalize you for the same thing again. So the diagram shows the labeling for A and B, but we want you to include correct labels and shading of the different chromosomes, except for labels A and B. So please don't label A and B because you've already been given marks for that in question 223. Right, so we need to draw a labeled diagram to represent the cell as it would have appeared in metaphase 2, and we're going to look at this on the next page. So here you can see the memorandum answer. Candidates were unable to work out the number of chromosomes, their arrangement, and their genetic configuration in order to successfully produce the correct drawing. And this is something that has been coming up in NSC exams for many years now, is the ability to draw different phases of meiosis. And in last year's NSC teaching tool, we also provided revision in this regard. So many simply just redrew the diagram from the question paper and included labels that they were not asked to include. So if we look at the marking guideline, the first mark was awarded for the correct position of the chromosomes, forming a single row on the equator. The diagram in the paper showed anaphase 2, and we had the chromatids separating. So all we had to do is to pull them together, put them together to show the previous phase before they separated. So learners could have redrawn the diagram and just put the 2 chromatids together to show that they are in the center. The number of chromosomes should have been 2 because remember that the division of the number of chromosomes already happened in meiosis 1. Then the shading of the chromosomes just needed to be copied from the diagram as is, again just moving them together. 1 shaded chromosome with an unshaded tip, 1 unshaded chromosome with a shaded tip, and then any 2 other correct labels besides spindle fibers and centrioles. That was A and B. So they could have provided a label for cell membrane, cytoplasm, chromosome, centromere, or chromatid, and gotten 2 marks for that. So this is a vital skill, and it's something that learners are still struggling with, even though it has been mentioned in many diagnostic reports. So let's practice this. So let's start with an easy one, which is just a verbal instruction to draw an animal cell during anaphase of mitosis, and it has to have 4 chromosomes. So that's a very easy one. You start by drawing an animal cell, could be round, could be slightly oblong. Then you have 4 chromosomes, And because this is anaphase, the chromosomes need to be moving apart from one another. So you can see they're forming their little boomerangs, and the spindle fibers are shortening, pulling them towards the poles. And then you can add any labels as requested: centromere, cell membrane, cytoplasm, unreplicated daughter chromosome, also chromatid would have been accepted, and centriole. So the mark allocation for this would be the correct phase having been drawn. So it's anaphase. You can see that chromatids are moving to the poles, that there are 4 chromosomes present and not any other number. 4 chromosomes that are split, so 4 chromatids moving to each pole. No genetic material has been exchanged because this is a drawing of mitosis, not meiosis, and any 2 correct labels as provided. So that's a very easy one that they should have been able to draw in grade 10 already. So let's try another. So this is from the NSC paper 2015. Draw a diagram providing the position of the alleles to show the structure of the chromatid Y after crossing over has occurred. So learners basically just needed to draw the end result of this process. So looking at the memorandum, they basically just want you to draw structure Y. So structure Y is one chromatid, and you can see that it carries genes D and R, but then at the point where B would have been located, a chiasma has occurred. So there's going to be an exchange of a chromatid segment. So Y is an unshaded chromatid, and it has exchanged a shaded section with a different chromatid that contains the gene small b. So ultimately, your chromatid Y needs to look like this on the memo, with a big D gene, a small r gene, and then a shaded section of the B, small b gene. Mark allocation was to actually have only chromatid Y represented, so the large white top and a smaller black bottom. And then 1 mark was awarded for indicating the alleles correctly, so writing the letters D and R and B in the correct spaces, in the correct formatting as a capital letter or a lowercase letter. Okay, so let's try another one. Let's ramp up the difficulty. From an NSC paper 2015, paper 1, when they were still testing meiosis in both papers, so please note that this is not the case anymore. Draw a diagram of the cell below as it would appear during anaphase 1. Okay, so first things first, you have to identify what phase is being shown currently. So we can see chromosomes that are not in pairs, which tells me we are not in the first phase. We're not in meiosis 1. We can see chromosomes that have spindle fibers on either side of their centromere, which again confirms to me that when they split, if the spindle fibers shorten here, it's going to split the centromere. So we're definitely not in meiosis 1. We're somewhere in meiosis 2. And then we can also see that genetic material has been exchanged already. We can also see that the chromosomes are lying on the equator of the cell, and that arrangement should hint that this is actually showing metaphase 2. So once we have established what phase we are looking at, now we go back to the question. It asks us to draw this same cell as it would have appeared in anaphase 1. So we have to go all the way back to anaphase 1, the first part. Okay, so in anaphase, we want to see a separation of either chromosomes or chromatids. Because this is anaphase 1, it would mean that chromosomes are separating in terms of their pairs. So these 2 chromosomes, we have to actually draw the pairs that they used to have been attached to. Now, if we look at the shading of the 2 chromosomes, we basically just need to invert that to draw their chromosome counterparts in their pairs. So for this chromosome, we're going to draw another little butterfly shape, and we're going to shade all the white parts black, and the black part stays white. Same we're going to do for the other chromosomes. So we're going to draw a little butterfly shape, and we're going to shade all the white parts black, and the black segment stays white, because that's where the exchange has happened. Right, so now we've drawn the counterparts, the 2 chromosomes in their pairs. Because we have to draw anaphase 1, we need to move the pairs apart from one another, so there needs to be a gap in between them. And we need to have spindle fibers attached to each of their centromeres that is actually shortening and pulling the pairs apart. So let's look at the actual memorandum for this. Okay, so the little A man says, first you must identify that the phase you have been given is metaphase 2. Then you must work backwards to anaphase 1. So if there are 2 chromosomes present in metaphase 2, then there have to have been 4 present in anaphase 1, because remember that we're still in the process of halving the number of chromosomes. The chromosomes in your drawing must also show complementary shading to the ones that have been given. So the mark allocation was 2 marks for drawing 4 chromosomes that are present, so double the number of chromosomes, not just drawing chromatids. Then 1 mark for showing the separation of the genetic material. There's a clear space between the chromosomes, so it's actually indicating anaphase, and it can't be mistaken for having drawn metaphase. Then the correct variation is shown in the chromosomes, so a learner has shaded the chromosomes complementary to the ones that have been provided. And this is really, really a difficult skill, so it's important to keep practicing it with learners. So let's try another one. So this one is from the 2014 NSC March Paper 1. Again, we don't assess meiosis in paper 1 anymore, so this is from before that change. It says the diagram below shows a cell in metaphase of meiosis 1. So it tells you the phase. This is metaphase of meiosis 1. Draw a fully labeled diagram to represent one of the resulting cells at the same phase of meiosis 2. So this is easier in the sense that you're working forwards and not backwards. And that's what makes the previous question a little bit more tricky. So considering that this is metaphase 1, we need to move to metaphase 2. So metaphase 1, what we're going to see during anaphase 1 is that these chromosomes are going to move to the pole. So these 2 are going to move to this side, and these 2 are going to move to that side. So I'm going to draw 2 cells to represent my cells at the end of meiosis 1, and I'm going to copy the shading of those 2 chromosomes to the top, and I'm going to copy the shading of these 2 chromosomes to the bottom cell. And that's basically what you had to do, because if you then move forward, you think prophase, no exchange is going to happen, the nuclear membrane is just going to disappear, and then we're going to get to metaphase 2, which basically shows you in the memorandum that you have those 2 exact options. You're going to take these 2 chromosomes that are highlighted in red, and you're going to move them to one cell because they're going to move to one pole during anaphase 1. The 2 in blue are going to move to the other pole during anaphase 1 following this picture, and therefore you can draw them in their own separate cell. So the question asked for a fully labeled diagram to represent one of the cells resulting in metaphase 2, so you could have drawn either of these. Mark allocation was 1 mark for a caption. It's very important to always include a caption. Even if in the previous examples no specific mark was awarded for the caption, learners must always add a caption to a graph or to a table or to a diagram. So this is a cell in meiosis 2. They could also have said a cell in metaphase 2 because they've identified the picture as being metaphase 1. 2 chromosomes are shown within each of the cells. The alignment of the chromosomes got a mark because obviously they have to represent metaphase, so we have to see a single row of chromosomes in order to identify it as metaphase. And then the correct appearance of the chromosomes, so having to just redraw those, and any 3 correct labels, spindle fiber, chromosome, chromatid, centromere, were all options there. Okay, so this is a lot of revision, and it's something that needs to be done in even more depth. But we don't have enough time in this document, so we're going to return home to the question. Right, so we continue to the next question. 2.3 has to do with blood grouping. It says that the table shows the percentage of blood donors for each of the blood groups in South Africa for 2018. So we have the blood groups O, A, B, and AB, and we have the percentage donors shown in the second column. So according to the data, which is the most common blood group among the donors, that's just simply going to the table and seeing which blood group has the highest percentage, and that's O. Question 2 asks to name the type of dominance shown by the inheritance of blood group A. So blood group A is an example of inheritance for complete dominance, because to have blood group A, you only have an A allele, and then the second allele could be an O, which is recessive, or an A. So it's definitely a complete dominance that you can only have blood group A if you have at least one dominant A allele, and the other allele should be recessive or dominant A. Question 3 says to explain how it is possible for a man with blood group A and a woman with blood group AB to have a child with blood group B. So what I would have done in this case is to draw a Punnett square to kind of organize my thoughts, and from there I would provide the explanation. So you can't just draw a Punnett square, it's asking you to explain. So let's draw the Punnett square. This side is the man, he has blood group A, and this side is the woman, according to the question, who has blood group AB. Now, obviously, gender does not affect your blood group, but for the sake of this question, we need to explain what the man's genotype would be and what the woman's genotype would be. So for the woman, her genotype is pretty straightforward. Because she has blood group AB, which is determined by codominance, it means that she can only have the genotype IA, IB. Anything else would mean that she doesn't have the phenotype AB blood group. The man, however, is a bit of a problem. We need to figure out, does the man need to be heterozygous or homozygous in order to produce a child that is blood group B with this woman? So if the man was homozygous dominant for blood group A, he had 2 A alleles only, then you could see no child with blood group B would result. So this means that the man has to be heterozygous for blood group A. He has to have 1 A allele and 1 I allele, a small I, which designates blood group O, the recessive allele. If that were the case and we cross them together, we see that there's a 25% chance of having a child with blood group B. Now we need to put this Punnett square into words. So you're going to say, the man has to be heterozygous for blood group A. Or you say his genotype needs to be I, capital A, small I. The woman's genotype can only be IA, IB. The child then inherits the IB allele from the mother, and the recessive I, small I allele from the father. So therefore, the child will be heterozygous for blood group B, or in other words, the child's genotype will be I, big B, small I. So candidates referred to the inheritance of blood groups instead of the inheritance of the alleles. So they said that the child inherits the blood group B from the mother, instead of saying that the child inherits the allele for blood group B from the mother. They described a dominant blood group instead of a dominant allele. So we have an IA allele which is dominant, or an IB allele which is dominant, but it's not that blood group B or A is dominant. They failed to describe the inheritance of the alleles from both parents. That was the whole point of the question, because it says, how is it possible? Explain. So you need to tell me, where does he inherit what from which parent to have blood group B? And they used the incorrect notation for the alleles. Then question 234 asks them to plot a bar graph to represent the data in the table, and the memorandum is provided on the next page. So here we can see this is a bar graph. There are spaces in between the bars, and learners had to use the data on the table to construct it. So the marking guideline provides a mark for the correct type of graph that has been drawn, a bar graph, and that's always something I told my learners is, even if you don't know how to plot a graph, write a heading, draw 2 axes, put names on them, and actually just draw a straight line if it's a line graph, or draw 2 bars if it's a bar graph, because you could get 1 mark at least for drawing the correct type of graph, regardless of whether the content of the graph is actually correct. The graph caption also got a mark. If it had both variables in it, they needed to have something about South Africa, the percentage of blood donors for 2018, so those are things that needed to be included. Then the axes labels, X and Y, had to be correct for 1 mark, meaning that blood groups was on the X-axis and percentage donors on the Y-axis. The scale received 1 mark, which means that for the Y-axis, you needed to have a correct scale where, in this instance, the memo goes up in percentage intervals of 10. So learners could have obviously made that a different percentage interval, but it needed to be consistent. And then also on the X-axis, the scale here refers to the width between the bars. The biggest mistake learners make with bar graphs is often not drawing spaces in between them at all. So the bars are right next to one another, which then makes it a histogram. Or alternatively, the space between the bars are not of equal width. They just draw them freehand instead of actually using a ruler to make sure that there's a 1 or a 2 centimeter space in between each. And then the plotting of the coordinates from the table received 2 marks in total. Some notes is that a histogram or a line graph, if one of those were drawn, learners only lose marks for the type of graph and the scale. And then if they inverted the axes, they transposed it, meaning that blood groups was on the y-axis and percentage donors on the x-axis, they can get full credit if the axes labels are also swapped and the bars are drawn horizontally, so they're running in this direction. If the labels are not corresponding, then they lose marks for the labels in the scale, and you have to check that the plotting is actually correct for the given label. But yeah, we'll hope that learners actually don't do that at all and do a correctly proportioned and directed bar graph. Candidates lost marks for unequal spacing between the bars, as I've mentioned, and the incorrect use of the scale on the x-axis and an incomplete graph caption. So let's practice writing a few graphs based on previous exam papers. So this one comes from 2019's NSC June paper 2. It shows you a whole spiel about the average group of students observed the mating calls of a population of frogs at a local dam, which has recently become louder. And then they give you the results in the table showing you the year from 2006 to 2011, the average loudness of traffic noise and how this corresponds to the loudness of the mating calls. And then question 5 of this asked them to draw line graphs on the same set of axes to show the change in the average loudness of traffic noise and mating calls for the period of 2006 to 2009. So this is going to be a line graph. We're going to have 2 lines, one for average loudness of traffic noise, one for the loudness of mating calls. We can see that there are units, decibels provided. And we're going to have years on the one axis from 2006 to 2009. So the way that tables are always structured is that the independent variable is mentioned first. So year is our independent variable, and that's going to be on the x-axis. And then we're going to have these two, the average loudness of traffic noise and the average loudness of mating calls, are going to be the lines that we're going to draw. So the average loudness is going to have to be on the y-axis, and that's measured in decibels. In terms of the caption or heading of the graph, we basically need to describe all the variables involved. So what we're looking at here is we're comparing the loudness, the average loudness, you could have said, of the traffic noise and mating calls of frogs over the period of 2006 to 2009 in this particular area. So you got marks for actually having the dependent and independent variable in your caption and creating some form of a relationship between them. So the mark allocation, very typical, is that firstly, the type of graph got a mark. So you had to draw line graphs, 2 of them on the same set of axes. There had to be a key because there are 2 different line graphs. You had to show us which line is mating calls, which one is traffic noise. The title had to include all 3 variables, meaning the 2 dependent variables, the mating call loudness and the traffic noise loudness, and then also the independent variable, which is the time period. The scale for the x and y axis got 1 mark, so that, in other words, there was a consistent and correct scale. Labels, that you had years on the y axis and average loudness in decibels, including that unit on the y axis, and then plotting also received 2 marks. Let's try another one. 2017's March paper, it says study the table below that shows the change in antibiotic resistance in 3 strains of bacteria over a period of 20 years. And that could just as easily have been a graph caption, so you'll see that that corresponds to the memo. So again, this table is arranged with the independent variable first, which is the years. Then we have the antibiotic resistance, which is the dependent variable, and we're looking at it in 3 different bacterial strains. So it asks you to draw a line graph once again to show the development of antibiotic resistance only for the MRSA strand. So we're only going to be looking at those 2 columns. If we look at the memorandum, we see that you have your line graph there being plotted from 1981 to 2001, so all the data, but only for the MRSA strand. We have on the y-axis the antibiotic resistance in percentage for the MRSA strand, and then on the x-axis we have the years. And if you look at the caption, it says changes in the percentage of antibiotic resistance in MRSA from 1981 to 2001, or over 2 over 20 years. And that's very similar to what was in the heading or the stem of the question, which says the change in the antibiotic resistance in 3 strains over a period of 20 years. So the only difference is that we're going to say change in antibiotic resistance, percentage of it, in one of those strands, MRSA, over a period of 20 years. Again, the marking guideline has a very typical layout. You get 1 mark for the type of graph that's correct, 1 mark for the caption or the title with all the variables included, 1 mark for the label and scale of the y-axis in this case, 1 mark for the label and scale of the x-axis, and then the plotting of the points received a maximum of 2 marks. So let's try another one. It says lizards of a certain species on an island are usually brown in color. A mutation in one of the genes for body color resulted in red and black lizards. And then they go through a whole story about an investigation that was conducted into the relationship between the color of the lizards and the survival rate on the island. So what we see in the table here is the color of the lizards. That was our independent variable, whether they are brown, red, or black. And then the percentage of each color in the population as it survived is what they measured. So that depended on their color, their survival rate depended on their color, and that is our dependent variable. Then the question asked them to draw a bar graph to compare the percentage of the brown and black lizards in the initial population and the 30th generation. So we want to see how the brown and the black lizards changed in terms of their survival percentage from initially to the 30th generation. So if we look at the memorandum here, we see that there was one of 2 representations of this graph. Learners could have either made the x-axis the generation or the color of the lizards. So I would have opted for the second one just because that's the way that the table is actually organized. So let's focus on that one for now. So we have the independent variable, which is the color of the lizards on the x-axis, brown and black. And then the percentage of each color in the population is on the y-axis. That is our dependent variable. And then we're going to again be drawing 2 bar graphs on the same set of axes because we have the initial population for the brown lizards and the black lizards. And then we want to also see how that changed in the 30th population for the brown lizards and the black lizards. Alternatively, if learners chose to do it a different way around, they could have made the key brown or black and then put the initial and 30th population, in other words, the generation, on the x-axis. So here again, you get 1 mark for a bar graph that has been drawn, 1 mark for the title of the graph, which in this case is a comparison. We're looking at a comparison between, if we look at the second graph, the percentage of brown and black lizards in the initial and 30th generation. The 2 variables are mentioned there. And then the correct label and scale for the x-axis, the correct label and scale for the y-axis, and then drawing of the bars, meaning the plotting of the bars has been correct up to 2 marks. So here are then a few examples of how to practice drawing graphs, and you can do this by adding even more examples from past papers. But for now, let's continue home. Question 2.4 is on a pedigree diagram. It says muscular dystrophy is a genetic condition that causes muscles to weaken over time. It is caused by a recessive allele on the X chromosome. That's a very important statement. They give you the genetic annotation, so you immediately know that this is an X-linked recessive disorder. The dominant allele results in normal muscle formation. The pedigree diagram shows the inheritance of muscular dystrophy in a family. So when confronted with a pedigree diagram, step 1 is to first unpack the diagram, to add everything you can to it, and then go to the questions. With X-linked recessive disorders, we start by first indicating the genotype for all males that have the disorder. And we start with them because their genotype is known. A male has an XY gonosome pair. The Y chromosome has no genes that it's carrying in relation to the disorder. The X chromosome, having only one X chromosome, means that if anything is wrong or defective with that X chromosome, then the male will be expressing that disorder in their phenotype. So a male with muscular dystrophy has a known genotype as Xdy because we know that their only X chromosome will carry the abnormal recessive allele. If they had a normal a dominant allele, they wouldn't have muscular dystrophy. And that's the difference between men and women when it comes to X-linked recessive disorders. Because females have two X chromosomes, there is a greater chance that if one of them is defective or abnormal, that they would have a normal, healthy, alternative X chromosome to mask that one, to dominate the recessive allele. Males do not. So males on this pedigree diagram with muscular dystrophy is then individuals 4, 7, and 8, and we can add their genotypes to the pedigree diagram. The second step is to identify all the unaffected males because their genotype is also known. A male can only be unaffected if there is nothing wrong with his X chromosome in the case of an X-linked recessive disorder. Because he only has one X chromosome, if he carries a defective or abnormal allele on it, he will exhibit that in his phenotype, the disorder. So we know that males without muscular dystrophy have a genotype X big D Y, and we can then add those genotypes on the pedigree for individuals 1, 3, and 6. The third step is to identify any females that have the disorder. So in an X-linked recessive disorder, it's basically easy to identify the female's genotype as well, because if they have the disorder, both their X chromosomes will carry the abnormal recessive allele. Now, in this pedigree diagram, there are no females that have muscular dystrophy, but in the case that there was, their genotype would have been X small d, X small d. Now, the last step is to worry about females that do not have the recessive disorder, because their genotypes could either be X big D, X big D, so they're homozygous for the healthy, normal alleles, or they have an X big D, X small d combo, which means they're carrying the recessive allele and they can pass it on to their offspring, but phenotypically you cannot see anything wrong with them. They are not expressing that disorder phenotypically. So the easiest way to identify these two types of females is to look for any mothers that are normal or healthy and don't have muscular dystrophy, but that have produced sons that do have muscular dystrophy. Because we know that a female of that nature, a mother who is healthy phenotypically but produces affected sons, that mother has to have carried the recessive allele. Why? Because males inherit their X chromosomes from their mother. So the only way that a son could have muscular dystrophy is if his mother carried the recessive allele and passed it on to him. So in this instance, we know that is true for individuals 2 and 5. So both females on this pedigree diagram, numbers 2 and 5, will be carriers for muscular dystrophy. They have a heterozygous genotype because they are phenotypically normal. We know that their one X chromosome with the dominant allele that results in normal muscle formation is going to mask or dominate the recessive allele for muscular dystrophy that they're carrying on their other X chromosome. So once we have all the pedigree diagram genotypes filled in and we know exactly what's happening in this family, then we go to the questions. Okay, so how many offspring do individuals 1 and 2 have? So if we look at the pedigree diagram, we see a marriage line between 1 and 2, and then we have an offspring line and a sibling line between them. So these two individuals had 3 children. They had 2 sons and 1 daughter. Then give the phenotype of individual 1. Phenotype is always easy to provide in a pedigree diagram because the key gives the phenotype, and you can really just copy and paste. So we see that individual 1 is a square that has not been shaded, and that, according to the key, is a male without muscular dystrophy, and that's where you got your mark. Genotype of individual 2, we've already determined by going through a whole unpacking of the pedigree diagram, is that it is a female that has genetically been carrying muscular dystrophy gene. So her genotype is an X big T, X small d, which means that she's phenotypically normal in the sense that she's a female that does not have muscular dystrophy, but genetically she carries muscular dystrophy as a recessive allele. Question 2.4.3 says that we must explain why more males than females are likely to have muscular dystrophy. So this is a very typical question when it comes to X-linked disorders, and learners can kind of memorize a standardized response to this, which has to do with the fact that men only have one X chromosome, and then to explain how that one X chromosome means that they are more likely to inherit a recessive disorder. So the memo says any of the following 4 marks. Males only have one X chromosome, so they only need one recessive allele on an X chromosome to then have muscular dystrophy or that particular disorder. The X small d allele on a male cannot be masked by a dominant allele because they only have one X chromosome. On the other hand, the question says we must explain why it occurs more in males than females. So let's compare that then to females. Females, on the other hand, have 2 X chromosomes, which means that they always have at least one X chromosome that can mask the recessive allele. For a female to have muscular dystrophy, she needs to have 2 recessive alleles. And in females, the dominant allele on one X chromosome can easily mask the recessive allele. But in males, it's a case of you only have the one X chromosome, so if anything goes wrong with it, unfortunately, you're stuck with that disorder. So question 2.43, candidates were unable to explain how the presence of recessive alleles on the chromosomes of males and females could result in the inheritance of a disorder. Mostly, they only stated the type of chromosome. So they said males have XY chromosomes and females have XX chromosomes, which didn't receive any marks. They failed to state that males only need one recessive allele to have a disorder when it is linked to the X chromosome, whereas females need to have 2 copies in order to have that disorder. They were also unable to explain the inheritance of the disorder in terms of the masking and dominating of recessive alleles. Question 4, parents 5 and 6 plan to have another child, use a genetic cross to show the percentage chance of having a child with muscular dystrophy. So having established the genotype of individual 5 and 6, we can now easily do this genetic cross. Individual 5 is a female without muscular dystrophy, and individual 6 is a male without muscular dystrophy. From our analysis of the pedigree diagram, we know that the female is heterozygous, in other words, she carries a recessive allele for muscular dystrophy, whereas the male, being a male without the disorder, can only have the genotype of a healthy male, so X big D Y. Then you're going to do meiosis by separating the gametes. So we have X big D, X small d for the female, and we have X big D, Y for the male. Then we draw a Punnett square to represent fertilization, and we put these alleles together. Then for the F2 generation, we summarize the phenotypes that we see from the Punnett square. We see that we have a 50% chance of having a female without muscular dystrophy. Then we have a 25% chance of having a male without muscular dystrophy, and a 25% chance of having a male with muscular dystrophy. Now the question says, use the genetic cross to show the percentage chance of having a child. So not a son or a daughter, but a child in general of having muscular dystrophy. So if we look at the entire Punnett square, there's a compulsory mark that was awarded for saying that in total, there is a 25% chance of having a child with muscular dystrophy. So note that the mark allocation for genetic crosses like this is very stock standard. Because this is a genetic cross layout and learners need to follow a layout like this, or you can also see the alternative. The alternative layout is doing a line diagram cross. The mark allocation still remains the same because this is the second generation of parents. So the parents 5 and 6 planning to have another child. So it's a P2F2 cross. And then we get 1 mark for writing fertilization and meiosis in the correct places. We get 1 mark for giving the correct phenotype as described in the pedigree diagram, the genotypes, 1 mark for the correct gametes, and 1 mark for actually crossing the gametes correctly to produce the genotypes of the F2 generation. Then 1 mark for writing out the phenotypes, and I've mentioned that compulsory mark to actually answer the question that was originally stated. So the diagnostic report says that candidates lost marks for the incorrect wording of the phenotypes. So they only wrote has muscular dystrophy without muscular dystrophy. They didn't add the gender So very important when it comes to sex-linked inheritance. Gender needs to be added to your phenotypic description because it determines whether you have the disorder or not. It's linked to your gonosomes. And more importantly, learners were given a pedigree diagram with a key that actually showed how to word the phenotype. Male with muscular dystrophy, female with muscular dystrophy. So it's very important that learners actually remember for sex-linked inheritance that gender needs to be part of the phenotype. Some also use the word fusion instead of fertilization. This was mentioned in our first video on paper 2. Fusion is not an accepted term. Fertilization is what happens between gametes. Most candidates did not indicate that this was a P2 and an F2 generation. They wrote P1 and F1. Please note that this is the second group of parents and they're having another group of children. So P2, F2. So that concludes section B's question 2, suggestions for improvement of question 2. So a significant number of marks are allocated for meiosis, and a few tips on how teaching this topic may be improved include starting it with a thorough revision and baseline testing to see what learners remember from grade 10, to continuously highlight similarities and differences between mitosis and meiosis, meiosis 1 and meiosis 2, and meiosis 2 and mitosis. So that single-page summary in this document would be a vital thing to print out, have your learners paste in their books, and work back to it the whole time, constantly reference it. When describing the behavior of chromosomes during meiosis, be specific as to whether you are referring to a homologous pair of chromosomes or a single chromosome. Note that all phases of meiosis has to have a number 1 or 2. Interphase occurs between meiotic divisions and does not require the number. Do more exercises to identify the specific phases of meiosis based on diagrams, and be able to talk about or identify the preceding steps or the following ones. Practice the drawing of chromosomes and chromatids using shading and show the effects of crossing over an independent assortment. And we've provided practice for this in regards to question 2.2 and on page 46 of this document. The correct genetic notation for blood groups was also reported in the previous diagnostic report, but it still continues to be a challenge. Learners continuously talk about blood group A being inherited instead of the A allele being inherited. So here is just again a summary notation of the phenotype that's described as blood group A, B, or O. The genotypic notation, which is the I allele with the letter A or B attached to it, or in the case of O, 2 recessive small I alleles. And then to describe the genotypes, the individual is homozygous, it has 2 dominant alleles, or the individual is heterozygous because it has 1 dominant and 1 recessive allele. And in the case of blood group AB, it can only be heterozygous because it's formed by 2 co-dominant alleles. Learners are competent at doing genetic crosses to establish the possible phenotypes in the offspring, but they struggle to articulate inheritance patterns when asked for explanations. And one way in which I can really advise teaching this genetics is to, whenever you have a pedigree diagram, to actually go through the inheritance pattern of it before you tackle any questions, and to talk with learners about this individual receives his X allele or his X chromosome from his mother, so he's inheriting that allele from her, or this individual is inheriting the capital D allele for the dominant trait from his father. So to actually talk about the inheritance patterns as you unpack pedigree diagrams. Then they provide a possible criteria to follow when you're answering a question like this is to first establish the phenotype of the offspring. So if we think about the question we had in this paper, they said that the child has to have a blood group B. So the phenotype is a blood group B. Formulate the genotype of the offspring. This means that the child should either have a dominant IB allele combo, so he's homozygous dominant, or the child can be heterozygous, IB small I. Give the genotype and phenotype of the parents. So they said that the one parent had a blood group AB, which means that their genotype notation can only be IA, IB. The other parent had blood group A, which means that their genotype could either be IA, homozygous dominant, or IA small I, heterozygous, and then indicate which allele was inherited from each parent. So to produce a child with blood group B, the one parent, which was IAIB, had to provide the B allele, and the blood group A parent had to provide the recessive O allele, because otherwise that parent had no B allele to provide. So that's just an example of how you could formulate your answers. Then, to master the skills of drawing graphs, this was not only revised in this diagnostic report and teaching tool, but also the one from last year in more detail. Learners struggle most with writing a complete caption, which we did practice in this teaching and learning tool. It must mention both variables and any other specific details that have been provided. And please refrain from using the term versus or VS in your headings. I always told my learners this is not a WWE match. It's not length taking on, nutrition, length versus nutrition. There is some other relationship between the two. So please don't make it a WWE match.
If you found this video useful, give it a like and subscribe to our channel so you don't miss any new episodes. Follow us on Instagram or Facebook to stay on top of the latest TAS news and launches. So that's it for now from The Answer Series, your key to exam success.